After studying this module, you shall be able to know about human osteology. You will learn about biological identity. You will be able to identify the age at the time of death. Forensic osteology is a sub-discipline within forensic anthropology and principally concerns the analysis of human skeleton for medico-legal purpose. A need for this most frequently arises following an investigation of human remains resulting from unexplained natural death, homicide, suicide or mass disaster, or following the allegation of war crimes or genocide. However, with increasing frequency, the forensic osteologist may be asked to provide assistance to the forensic medical examiner regarding the confirmation of age of living subject for the purpose of both judicial accountability and appropriate immigration status. Whereas the task performed by a forensic osteologist can be extremely varied, only the more traditional role will be discussed here from the perspective of analysis of a human skeletal remain. Upon the discovery of a skeleton, the investigating authority requires confirmation that the remains are indeed human. Frequently, non-human bone is presented for identification, and occasionally what appears to be human may not in fact the bone, as it is not uncommon for investigators to encounter inappropriate disposal of polymer anatomical replicas. The successful microscopic identification of human bone will depend heavily not only on the experience of forensic osteologists but also on which elements of the skeleton are present. Small, relatively featureless sections from the shafts of the long bones or fragments of rib too difficult to distinguish from animal remains, particularly those of pig or sheep. Understandably, animals that are significantly smaller or indeed larger than a human being are often readily differentiated. Although it is not essential for forensic osteologists to be skilled in comparative vertebrate osteology, they must be sufficiently familiar with each and every aspect of the adult and juvenile human skeleton to be competent at eliminating non-human material. It is an expensive mistake if police investigation is initiated on the misidentification of animal bone. Now, let us see biological identity. There are four basic categories associated with biological identity, that is sex, age, at death, stature and ethnic origin. Each of these taken either in isolation or collectively allow a preliminary picture to build regarding the possible identity of the deceased and permits targeting the specific aspects of a missing person register. The accuracy with which each favor will be determined will depend heavily upon which skeletal elements are available and also on the state of prevention. As general rule, the more fragmented the remains and the less complete the skeleton, the more difficult it will be realized full identification of the deceased. Therefore, it is essential that the remains are recovered by a forensic archaeological or archaeologist or appropriately trained recovery person to ensure maximal retrieval of potential evidence. If there is multiple burial of commingled remains or indeed a fragmentation perhaps through explosive disruption, the first stage is attempt to reconstruct the body of the individual through assignation of individual elements. This serves to establish a minimum number of individuals represented. At this point, it becomes important to assign a bone confidently to be the correct side of the body and also identify its correct position within a skeletal sequence. For instance, if two left capitates are found, there must be at least two individuals present, which is equally true if two left third ribs are encountered. Therefore, it is essential that every element presented is identified and striated where appropriate. Other factors that may assist in separating bones of a specific individual are the relative size of the bone, discernible pathologically conditions and their color or prevention stature. Although the latter two can vary within the same skeleton if the element becomes separated and exposed to different physical environmental conditions. The state of skeletal development can be used to separate adult and juvenile individuals with relative ease, but the ability to separate commingled juveniles depends on the detailed knowledge of the osteology of immature bones. For example, the presence of separate pars laterals the occipital bones together with the distal ulnar epiphysis and distinct iliac thrust will definitely identify the presence of at least 3 subadults, as these elements cannot coexist. Now let us see determination of sex first in adults. Because methods for age and stature estimation are sex dependent, sex determination is usually the first step on the biological identification process. Reliability of sex determination depends on the completeness of the remains and the degree of sexual dimorphism inherent in the population from which the individual originates. In general, there are two main morphological differences between male and female skeleton. Male bones are generally larger, more robust, and female bones due to attachment of the larger and thus more powerful muscle mass, larger body size, and a period of delayed pubertal growth compared to the female. Second, the male pelvis is primarily adapted to be a bipedal strutting gait, whereas the size shape difference of female reflect the biomechanical comprising between the efficient walking and modification necessary for passage of a large fetal head through the pelvic canal. It is generally accepted that the two most sexually dimorphic elements of the skeleton and the pelvis of the skull. Many workers claim an accuracy of sex prediction in the region of 90% from pelvis alone and 80% from skull alone. However, the values are optimal and they may be lower if the bones are damaged or indeed if the individual originates from population with a low level of sexual dimorphism. Generally, the shape of the male pelvis is high and narrow, whereas that of the female is wider, shallower, with relatively and often absolutely larger inlet and outlet dimension. These differences are most obviously reflected posteriorly in the shape of the greater stychic notch and relative proportions of the body and iliac of the sacrum, and anteriorly in the subpubic angles, the shape of the pubic bone. A wide, shallower, greater stychic notch is considered to be female characteristics and is greater width of the iliac of the sacrum compared to the width of the body. Probably the element of the skeleton that can offer the most reliable indication of sex is pubic bone. Owing to its thin covering of compact bone and its ventral position in the many-burals, it is unfortunately one of the part of the skeleton to survive inhumation least successfully. Although the morphological differentiation or determination of sex is the preferred method of analysis by expert, as it permits the multifaceted accumulation of information, it is not sufficiently statistically robust to justify or to satisfy the judicial requirements. The differences between the male and female skull become discernible at puberty when the male skull develops features that reflect sites of increased muscle attachment, whereas the female skull tends to retain its more pedomorphic features. In the male, the glabella and supraorbital ridges, mastoid process, and the nostril and malar areas become more prominent, in contrast to the female, where all these features are smaller and the forehead remains vertical, more pronounced frontal and parietal eminence. The orbits are more rounded in female and have a sharper margins and are larger relative to the upper facial skeleton. The male mandible generally has more prominent chin ridge, a more robust lower border, a greater body height, and more prominent muscle marking. The angle formed between the body and ramus is more acute in the males than in the female. Now, let us see in the cases of juveniles. As in the adult, the observation of sexual dimorphism have tended to concentrate on the pelvic and skull. Some researchers claim an accuracy of between 70 to 80% for certain measurements of the ileum and femur, but these figures have failed to be confirmed or retesting. Claims that the morphological differences in the surface of the sacroiliac joint could separate males from females have proved to be limited use in sexing, and some traits of the orbit and mandible show dimorphism if used under careful control condition but remains to be tested further on a population of known sex. At the present time, however, it has to be concluded that the juvenile skeleton cannot be sexed with any degree of reliability from morphological observation alone, although some slight sexual dimorphism may exist from an earlier age. It does not reach a high enough level to permit accurate sexing until pubertal modification have taken place. Some of the issues pertaining to the sex determination from both adult and juvenile skeletal remains have been addressed with some success following technical advances that facilitates the isolation and amplification of DNA from bones and teeth. The amelogenin gene responsible for production of one of the organic component of the enamel of teeth has sequence difference on the X and Y chromosomes, and a method utilizing molecular genetic techniques correctly identified 19 out of 20 skeletons of a known sex. At present time, there are still problems with contamination and degradation of DNA obtained from both bones and teeth, especially in certain burial conditions. However, the improvement of current techniques and development of new methods, sex typing from genetic material could be in future becomes routine in forensic cases. Now, let us see age at death. It is useful to divide age-related changes in skeleton into three different phases of an individual lifespan, that is the growth and development, equilibrium and senescence. The first phase is largely under genetic environmental influence and includes children and young adults who undergo changes that proceed in a relatively well-documented pattern at moderately predictable rate. The multiplicity of the composition of the juvenile skeleton offers an extensive array of growth related parameters from which to estimate age. Accuracy is therefore reasonably high. Once maturity is reached, the age-related changes in the adult skeleton vary quite significantly and more individual and population specific, especially at the stage when maturity proceeds towards senescence and subsequent degenerations. In this phase, factors such as health, status, occupation, nutrition and endocrine functions are principally determinants of degenerative status. In addition, most of the methods in use of adult aging rely on studies developed from the larger archaeological sample of unknown sex and age. When tested against samples of known age, this has cultured or resulted in systematic errors, the most important of which has been tendency to underage individual of over 40 years. In more recent work on a documented material, particularly following the Balkans conflict, the principle changing, although the genetic homogeneity of the material must be borne in mind before attempting to utilize the data or material of the different origin. Now see in juveniles. In juvenile age range, before cessation of growth in the height age may estimate from numerous markers from the teeth and skeleton, so that the estimate age bands are usually narrow than those of adults. Dental age estimates may be derived from the eruption of mineralization of teeth and are closer to the actual chronological age from those of the skeletal age, but the basis for this is not fully understood. A possible reason is that the development of all the dioecious dentation or a part of permanent dentation takes place before the birth is protected environment, whereas skeletal growth and development, although having a stronger genetic basis, is exposed for a longer to external factors such as the nutrition, socioeconomic status and even climate. Eruption is a continuous process by which teeth move from their crypts in the alveolar bone of the jaw to a full occlusion in the mouth. But many studies are confined to their emergence into the mouth, often wrongly referred as eruption. The presence or emerged teeth may be observed easily by osteologists to give rapid estimate of age, but increased accuracy can be obtained from evolution of calcification of the teeth. However, this necessitates radiographic analysis and comparison with defined stages of mineralization of both crowns and roots. This is a complex process that requires a considerable degree of experience and should be undertaken in conjunction with the forensic odontologist. Skeletal age can also be estimated from size and length of the long bones and the development state of center of ossification. When using the length of long bone shafts, accuracy will vary with period of life. Length in the fetus are closely related to the gestational age, and there are large amounts of clinical ultrasound data available for reference. Postnatal accuracy of age estimation decreases with increasing age as external factors start to affect the growth, and there is an individual and sex variation in height, especially during the adolescent growth spurt. Data also becomes a problem as the tables for comparison are based on systematic, non-repeatable cross-sectional and longitudinal radiographical surveys of children from at least 2 generations ago. The majority of these surveys were drawn from white middle-class people of European origin, but there are many populations for whom there is no adequate sample for comparison. It is also possible to age a juvenile skeleton from the development state of the primary and secondary centers of ossification. Most centers of skull, vertebrate and primary centers of long bones and their girdles commences ossification in the embryonic and fetal periods, whereas the majority of secondary centers develops with cartilaginous templates throughout the postnatal life. When a primary or secondary centers has reached a recognizable distinctive morphology, it may be used to advantages in aging. This critical point varies with each bone so that accuracy will depend on the skeletal elements are available. Most of the bones of skull, vertebra, ribs and major long bones of the limbs and girdles are recognizable from mid-fetal life onwards, but others do not reach a recognizable stage until later childhood. Now let us see young adults. Several areas of skeleton do not complete their growth until the second and third decades of life that are consequently valuable for estimating the age at death in young adulthood. This period extends from cessation of growth in height signaled by the fusion of all long bone epiphyses until the final fusion of all other epiphyses. This includes the jugular growth plate of the skull and postcranially, the scalar or the sacral vertebral bodies, the iliac and ischial epiphyses, the ring epiphyses of vertebrate bodies and the epiphyses of scapula, costal notches of the sternum and middle epiphyses of the clavicle. Now let us see the adults. Aging in the mature adult has a traditionally utilized 4 areas of skeleton: the state of fusion of the cranial statues, morphological changes in the pubic symphysis, sacroiliac joint and ventral aspects of ribs, closure of the ecto and endocranial aspects of the vault. Statues have proved very unreliable as age marker in spite of much work attempting to test and refine the original methods. This is unfortunate as the cranial wall survives inhumation probably better than any other skeletal element. Related to this, recent observation stated that the pits on the endocranial surface of the frontal and parietal bone that accommodate arachnoids granulation, granular fuoli, increases in number with age. Early results showed a high correlation with age, but the method still needs to be tested for reliability on a larger sample of age-documented individuals. Methods for aging from sternal end of the fourth rib also employ a phase analysis system, which is claimed to display an accuracy similar to that obtained for pubic symphysis and better than the aging from cranial statues. However, further testing showed that it was more accurate at determining age in males than in females and tend to underage individuals over 60 years. Apart from wide estimates of age, the disadvantages are that it uses one of the least well-preserved parts of the skeleton, and also requires the identification of fourth rib, which can prove difficult in a disarticulated, fragmented or disrupted skeleton. The method has been reported reliant on the previous experience of the observer. Used together with histological techniques on a documented sample, it showed a poor correlation with chronological age. Now let us see stature. The estimate of the height of an individual from skeletal remains can be, with certain exceptions, one of the most straightforward parameter to be established. The calculation of stature is based on relative proportion of different body parts, both in relation to each other and in relation to the overall height of the individual. Statures can be estimated with considerable ease and quite some accuracy from the long bones of disarticulated adult skeleton. The greatest accuracy will be obtained from undamaged bones of known sex and ethnic identity are utilized, as the height of the individual is both sex and race dependent. Most standard forensic texts contain regression equations from which adult male and female statures can be calculated, and recourse to these is recommended. Those formulae that use the femur and tibia have the lowest standard error of the estimating at these bones from integral part of an individual's height. However, when the preferred skeletal elements are not available, an attempt at the stature estimation may be made from metatarsal, clavicle or even fragmentary long bones, but accuracy will be declined rapidly with the use of incomplete bones. Again, it must be stated there are many populations for which no data are available. Now let us see the positive identification of personal identity. Once biological identity has been established, the forensic osteologist must attempt to personalize the information available so that individual identity is achieved. Many of the ways in which this can be done have been outlined in other chapters of this particular paper. Therefore, only a very cursory discussion takes place here in the sake of completeness. During the Asian tsunami, the three principal means of personal identity were achieved through dental records, fingerprints and DNA analysis. All of these required the pre-existing ante-mortem data set be available for comparison. When it is known that a person missing or deceased, then these records are passed to the investigating authority. However, when an unidentified set of remains are found, then dental records are of no value until a possible identity has been achieved. And it is common that no match is forthcoming from either national DNA or fingerprint database. Under these circumstances, the forensic osteologist must look to the other possible evidence within their means that may assist in the identification process. Now let us summarize what we have learned in this module. Forensic osteology is a subdiscipline within forensic anthropology and principally concerned the analysis of human skeleton for medico-legal purposes. There are four basic categories associated with biological identity, that is sex, age at death, stature and ethnic origin. Each of these taken either in association or collectively allow a preliminary picture to be built regarding the possible identity of the deceased and permits the targeting of specific aspects of missing person register. Other factors that may assist in separating the bones of specific individual are the relative size of the bones, discernible pathological condition and their color or prevention status. Once biological identity has been established, the forensic osteologist must attempt to personalize the information available so that the individual identity is achieved.